Hey class, this is Mr. Jackson here. This video is going to be a half review of week 5 where we're going over the cardiovascular system. Specifically in this video, I'm going to be talking about the conducting pathway of the heart from the pacemaker cell to the heart muscle tissue, the myocytes themselves. Okay, so let's review from last week. We remember that the heart has 4 chambers, so the top ones are the atria, the bottom are the ventricles. The goal is basically to pump the atria first, the blood from the atria to the ventricles, and then the ventricles are going to simultaneously contract right after that and pump the blood either to the lungs, through the pulmonary circulation, or through the body, the systemic circulation. But this is a highly coordinated process, and the way it's done is by action potential generation, nerve signals essentially. So what's going to happen is the SA node, the pacemaker cells of the SA node, are going to fire action potentials and basically stimulate the atria, the atrial myocytes, to contract first. Once it does that, at that point, the atrioventricular node, the AV node, will send its action potential down the node of His and then up to the Purkinje fibers and then depolarize the ventricles, which will then contract through the atria, either the pulmonary circulation again or the systemic circulation. But this is very coordinated because the SA node has to basically stimulate the atria first, and then about 0.14 seconds later, the AV node is going to get that stimulation and then fire itself so that it can depolarize the ventricles. So it's boom, 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 boom. Okay? This is what's happening on the microscopic level. So I drew on the left side, this is a pacemaker cell. So this would be found in either your SA or AV nodes, both. But I'm going to talk more about the SA node. Okay, so let's pretend this is the SA node cells. It'll be connected by millions of them, but this is just one pacemaker cell connected to the cardiac myocyte itself. This is the physiology of how this muscle basically gets told to contract. It's a multi-step process, so walk with me through it as we go through. So first off, the cells in the body, I've written a lot of minuses because the inside of the cells are usually very negative. When they are negative, they're considered to be at resting membrane potential. In the pacemaker cells, this resting membrane potential will be somewhere around negative 60 millivolts, give or take, whereas the cardiac myocytes will be more around negative 90 millivolts. So very low voltage, right? On the outside of the cells, though, they are very positive. So the exterior of the extracellular fluid around it are very positive. That's because you have ions surrounding these cells, such as, I'm going to draw this in red for you, such as sodium ions. So you've got a lot of sodium outside of your cells. So there will be some sodium up here too. Just drew a different color. There's some sodium up here. And there's also going to be calcium. So there will be calcium ions as well. And there will be other positive ions too, but these are the ones we're going to talk about today primarily. Okay, so if there's positive outside, negative inside, the positives want to flow in. Normally they cannot because there's a plasma membrane basically blocking off those charged ions from getting in. So there's this separation of charge inside and out. But in the pacemaker cells, there are literal channels called funny channels. These are leak channels for sodium. So if there's a high concentration of sodium outside the cell, low concentration inside, it's going to flow down its concentration gradient into the cell through these funny channels. So that's step 1, is that sodium will flow in through these funny channels. So sodium flows in through the funny channels. Well, think about it. If sodium flows in, it's a positive ion. So therefore, inside of the cell, it's going to start to become slightly positive as these sodium ions flow in. As it does that, it's going to go from about negative 60 millivolts to now about negative 40 millivolts once the sodium gets into the cell. Okay? So at negative 40 millivolts, this is called the threshold potential. At the threshold potential, the voltage-gated sodium and calcium channels will open. Okay? So, sodium just flowed in from funny channels. Now, these guys, these voltage-gated calcium and sodium are going to open wide. And something's going to happen. So, at that threshold potential, this gate opens. So this will be, what did I label it as? The voltage-gated sodium. And then the voltage-gated calcium is going to open as well. And you could probably assume what's going to happen now. If we have high sodium and high calcium outside the cell, they want to flow in rapidly. So the sodium will flow into its voltage-gated ion channel. So here comes the sodium flying in. And the calcium, I should have drawn this in blue. That. So the calcium is going to be in blue, and it's going to flow in as well. So now all these calcium ions are flowing in, all the sodium ions are flowing in, and we have this massive burst in positivity inside the cell. So now the cell is considered depolarized. So an action potential has been reached. So now the cell is very, very, very positive. So what were the steps? Well, threshold potential was reached. These gates opened, voltage gates open, and then positive ions flow in, and membrane reaches about positive 10 to 20 millivolts. This is considered an action potential when all these positive ions are flooding in. Now, what was our goal at the beginning? Well, our goal was to contract this muscle cell. And we haven't done a darn thing with this muscle cell. But if you look at the muscle cell, you'll notice that they also have voltage-gated ion channels, calcium and sodium. So we need to get some of the positive here into here so it can reach its threshold, open its voltage-gated channels, and then reach an action potential to contract. So how do we do that? These positive ions, we'll do step 4, the positive ions flow through the gap junctions that connect the two cells, thereby making this cell, this myocyte, more positive. So once some of those positive ions flow into the muscle cell, the cardiac myocyte, now myocyte reaches its threshold potential, reaches threshold, which means now our voltage-gated ion channels are going to open. So now my sodium one has opened wide, my calcium one has opened wide, and now all of the ions in this section are going to flow into the cell, into the myocyte. So now all my positive ions are flowing in, making this cell very, very, very positively charged because it's adding a bunch of positive ions. Now, this guy's depolarized now, so it's just reached probably, I think it goes up to like positive 20 millivolts. I could be wrong with that number, but it reaches a positive number. But we need it to contract, and it's not enough to just throw in a bunch of positive ions to contract. So once they reach the threshold and the ions flow in, this will be the next step, so positive ions flow in, there's going to be a thing called calcium-induced calcium release. Since calcium is flowing in, the calcium itself will trigger a storage bin called the sarcoplasmic reticulum, I'll just do SR, will trigger the sarcoplasmic reticulum that stores a lot of calcium to release its calcium. So now the sarcoplasmic reticulum is basically a modified endoplasmic reticulum within the cardiac muscle cell. When it gets stimulated by calcium, it'll basically explode with a bunch of calcium into the cell. And if you remember from the muscle unit, the muscular system, calcium allows the troponin and the tropomyosin to basically pull back, allowing actin and myosin to interact and then contract. Okay? So, the calcium allows for, I'll say actin and myosin binding, myosin, which leads to a muscle contraction. And now the muscle has contracted. But at the same time, we need to get it back to its resting state, right? The goal was to contract it, contract the ventricles to pump the blood, but then they also need to relax so that they can fill with blood again, right? So, what happens? Well, what happens is there are going to be a variety of pumps that basically reestablish the membrane gradient. So, pumps reestablish gradient, and it returns to its resting potential. So as soon as, for example, as soon as this pacemaker cell basically fires its action potential, gets really, really super positive, up to like positive 10 millivolts, it can then no longer be stimulated. Those pumps will be activated, basically pump all those positive ions back out of the cell, and actually kind of bring in some negative ones too sometimes. Sometimes, don't quote me on that. But if it pumps a lot of positives out of the cell, therefore the cell will become more negative and reach its resting membrane potential again. Okay, I'm not going to step through the process of that because I think it'd be a little too complex. But just know this whole process of how funny channels are always open, sodium flows in, allows voltage-gated ion channels to open up, positive ions flow into the pacemaker cell first, okay? Then, once it flows in, some of those positive ions will flow through gap junctions, going to the cardiac myocytes, allowing it to then open its voltage-gated ion channels. Those will flow in, trigger calcium-induced calcium release from the sarcoplasmic reticulum, bind to that troponin head, pull it apart, and then actin-myosin can then do its work and contract, and then pumps will finally reestablish the gradient once again. So I'll step to the side, I'm going to screenshot this, it's a very helpful diagram. And just to step back a little bit, these action potentials in these myocytes will be reached first in the atria. So these guys in the atrial myocytes will contract first, and then the AV node will basically be the pacemaker cell for the ventricles. It will then get stimulated, and then the ventricle myocytes will contract. So it's a very coordinated process. Atria, ventricles, atria, ventricles, atria, ventricles. Hopefully this was helpful. Drop any questions you have in the comments below. Be sure to like and subscribe. I appreciate it, and we'll see you guys soon.